Hello, everyone. So welcome to today's IQB seminar. Our speaker today is Dr. Haoda Fu. Let me give a brief introduction for Haoda. So Haoda is a research fellow and a enterprise lead for machine learning, artificial intelligence, and digital connected care from Eli Lilly and Company. Haoda is also a fellow of American Statistical Association. He's also an adjunct professor of biostatistics at Indiana University School of Medicine. Haoda received his PhD in statistics from Wisconsin Madison in 2007 and joined the Lilly after that. So since he joined the Lilly, he's very active in statistics methodology research. He has more than 90 publications in the areas such as Bayesian adaptive design, survival analysis, recurrent event modeling, personalized medicine, indirect and mixed treatment comparison, joint modeling, Bayesian decision-making, and rare events analysis. In recent years, his research area focuses on machine learning and artificial intelligence. His research has been published in various top journals, including JASA, JRSS, Biometrics, ACM, IEEE, JAMA, Annals of Internal Medicine, etc. He has been teaching topics of machine learning and artificial intelligence in large industry conferences, including teaching this topic in FDA workshop. He was board of directors. He was board of directors for statistics organizations and program chairs, committee chairs such as ICSA, ENAR, and ASA Biopharm session. And today, Haoda will talk about his recent research on the dynamic treatment regime and utilizing the machine learning and artificial intelligence approaches. So welcome, Haoda.
Okay, yeah, thank you for Steve and Stian for the invitation, and thank you for the Stian for the introduction. So for myself, I'm going to share my slides, and we can get started. And so, 1 second, share my screen. Before I was talking about I will make these slides as a slides presentation mode. But before I talk about these slides, I'm thinking that it may be good. I provide a little bit of background myself and Eli Lilly and companies, and so what is the research, what we are doing to give broad pictures before really and jump in some of the technical stuff. Because the purpose, I'm thinking one purpose of the presentation is to share what our research looks like, but that's only one of the purpose. I'm thinking another most important thing is to let people to get more familiar with Lilly and get more familiar with our research and to increase some interest for what we are doing and lay some foundation of our collaborations. I know this is the first time I come to the Rutgers to give the presentation, and certainly that may not be the last time. I will have more opportunities to work with you guys. So I'm still in my first page of the slides. I didn't mean to move my slides for the next maybe 10 to 15 minutes. I want to give a little bit of introduction. So start from Lilly. Some people know, I know some of the faculty members, some people know Lilly really well. Some people may not know Eli Lilly and Company. I'm from Eli Lilly and Company, and Eli Lilly and Company is one of kind of the top kind of maybe top pharmaceutical companies in the US based company, and we are in the Indianapolis area. One stories, and people may know some. I believe people must know some of the product from Lilly. I still remember about before I joined Lilly, I read the papers in the year 2005 or published in Nature or either published Science or Nature. They talk about the top 10 molecules really changed the human history. So they look at the past human histories, and there's 10 drugs that really changed the human histories. So among these 10 drugs, 3 are coming from Eli Lilly and Company. They talk about the Lilly's research. So the three molecules to demonstrate the Lilly history. The first one is the insulin. The insulin actually is exactly about 100 years ago. Lilly started to commercialize to develop research and working with Universal Toronto's and commercialize the insulin. So that is the first broadly commercialize the insulin product. Because of that, and it saves kind of millions of people's life. And so that's the first molecule really ranked change the human history, because before the insulin, there's no treatment for in particular for type 1 diabetes patient. If the case got a type 1 diabetes and the case will died, and people get a later stage type 2 diabetes, and there's no treatment for those patients. The insulin really changed the human body's life, and so that's the first molecule really changed the human history. So among these 10, the second molecules is antibiotics, the penicillin. So the Lilly is the first company to commercialize the penicillin and this antibiotics. Before this, there's many of the treatment kind of there are many of the disease is very difficult to cure before the antibiotics age. And the people also talk about the antibiotics, maybe the one of the most important molecules, most important drug for human history. And just because of that, they are increased at the average human being's life from about 40 years old to the past the Second World War to the 65 years old. They credit a lot for the discovery and commercialized of the antibiotics. So penicillin is from Lilly. The third molecule coming from Lilly is the Prozac. The Prozac is so the people have this anti-depression drug before the Prozac, and the people feel like the depression is a psychology problems. They may need to talk to more people, doing some exercise, but Lilly provided this treatment for the depression. So that demonstrates Lilly have the huge history of the for helping the patient to improve the patient outcome. Of course, there are many other products Lilly was working on. So, for example, people these days talking about this pandemic, and actually the Lilly finished one of the pandemic after the Second World War. So it's folios. The folio is a virus and causes many of the kind of causes some of the paralysis of the people, and even the president Roosevelt have these problems, and Lilly developed treatment finish the pandemic of the folios. And these days, probably people also see the news in early this week. Lilly also got FDA approved for the treatment of the COVID-19. So let's talk about Lilly, and our team. I'm currently leading the Lilly enterprise-wide machine learning, artificial intelligence, and algorithm development team. So we originally coming from the global statistics organization. And after that, people see the important to combine the data, analytics, and the technology together to have one organization. This organization want to have a broader impact for the analytical solution for the company. So that's why they form this so-called AADS. You can see I'm still in the first page of my slides. If you see my slides, they call it Advanced Analytical Data Science Organization. So that's currently I'm in this organization. In this organization, they have different directions. And I'm currently leading the enterprise-wide machine learning, artificial intelligence, and algorithm development team. So our team developed this machine learning algorithms and data science solutions to support for all enterprise level activities. We covered a few kind of important area for the company, which is we call the data rich area. So these 4 areas we covered is including the digital health. The digital health is how can we use a digital solution to helping the patients, including the isodalometers and the geoscopes and digital biomarkers. And how can we use this solution to improve the patient outcome? The second area is diabetes. We call it diabetes connected care. And diabetes connected care is how can we better use the insulin pump and the insulin to insulin pump and CGM and build the whole system to helping the diabetes patient. As you mentioned before, like Lilly have a long history to treating diabetes patients, started to commercialize the insulin product. And these days, about 50% of revenue is coming from Lilly diabetes business unit. and our team closely working with this group to develop the kind of general digital solution to helping diabetes patient. The third area is the manufacturing. The manufacturer is needed data analytics and machine learning algorithms to improve the productivity and the efficiency. The last area is really related to what we are going to kind of potentially collaborate together. Is the Lilly Research Lab. We call it LRL, the Lilly Research Lab. That the Lilly Research Lab have about 8,000 of researchers and about 4,000 PhDs. This size is really comparable, like one of the large, like the schools, and like many of the PhD and doing the researchers. They include some of the biologists, the chemistry, kind of the statisticians, and there are different disciplines that people work together and for the drug discovery development. One of the important areas that our team was working on either in the preclinical, in the drug discovery space. In the drug discovery space, there are two important problems. One important problem is the target identification. How can we find this the target that we can develop the drug? So the target identification. Once we found the target, another very important area is the molecule design. So it seems like we found the lock and we want to find the key. How we can design the key to open the lock? So that is the very important problems. This morning, I know I met with a few. I met with the faculty members, students, and this very active research area in this area. So if we double click this molecule, if we double click this molecule design, there are two subcomponents. One component is how can we use the molecule structures to predict the molecule property. So these are traditional, like called the QSAR work, the structure action relationship predictions. So we have been published a few papers in this area to use the molecule structure to predict the molecule property. And with that, we also develop some of the virtual screening platforms. And so once we have the molecule structures, and we can quickly to use the simulation-based screening and to see if the molecule can achieve the desired property. So that's a property prediction, and this property prediction under this the molecule design umbrellas. But once we can do better predictions, the next question is, can we really to use algorithm, machine learning algorithm, design a molecule? And people probably already saw many of these generative models, and they give one picture and they make this picture as a certain style of the artist, like paintings, and to design this, modify this picture a little bit, so this picture can better achieve the certain property, like mimic some of the artists. Like similar, like for here, if we have one molecule and we want to improve the solubility of this molecule, and how can we modify this molecule structure can achieve this property? So there's another important area that what we are working on. So to make a summary, so our team, our function focus on 4 important business area, and among these 4, the Lilly Research Lab is a really important area. Under the Lilly Research Lab, one important component is the drug discovery. In the drug discovery, have 2 sub-important problems: target identification, molecule structure design. Under this molecule structure design, we have 2 maybe the sub-effort. One is the property prediction. Another is how we can generate the molecule to better achieve the desired property. So that is the 4 business area our team is focused on. We hired people from different disciplines. In my team currently, we have like 11 people in my team, including myself. We have our skill set, this 11 PhD, we have some master levels and the data, some to support us. Among 11 of us, we have a different discipline. So some people have a training in the statistics, some people have a training in the computer science, some people have a training in the electronic engineering, computational biology, different training. By the way, we do have openings for the computational chemistry, computational biologists in my team. So if you are interested in some of the positions, if you happen to looking for a job and as a graduate student, so feel free to add me at LinkedIn so we can have some further discussions. And also this afternoon, I'm going to talk with a student. Lilly, the really good company, have a lot of history in the area. So that is kind of the research what we are doing under this. And we also have a 4 area kind of we have the 4 business area. We also have a 4 capability area that what we are building at this moment. And we hire people kind of from this computer scientists, people working on optimization with a different multidiscipline and to work on the areas. And the 4 areas that what we are working on is the first area that what we are working on is the recommendation system. How can we give the recommendation? The second area is automatic control. It's talking about the control theory and in the insulin pump, glucose pump. And the third area, the sequential decision making. How can we optimize the sequential decision making? But the last area is also very relevant for us is how can we better understand this non-structured data. Non-structured data, including this non-standard, non-structured data, including the image analysis. The image analysis, kind of the sequence data. Sequence data is like the natural language processing. And kind of this technology is very relevant for the current department, kind of the Rutgers. I'm talking with the students that are interested in how we can better leverage the deep learning technology, and to better understand the molecule structures, understand this RNA sequence and how they are kind of synthesized, relate to the protein structures, and how these protein structures from the graph point of view, from a 3-dimensional and kind of a point of view to connect it with the protein property, kind of the bindings properties, and to do a better prediction. So that area is also we are very interested in. So if we look at our focus business area and also look at our capabilities, so you can see in the molecule structure space, and the use the deep learning to predict the molecule structure, to generate the molecule structure. That's what we are really interested in. I know kind of currently the Steven and the Sijian's team have a lot of strength in this area and do a deep research in this area. That's why we are really interested in some of the potential collaborations and to moving forward in the area. And that's, I'm thinking, is one of the most kind of interesting point I want to talk with the department. And I know the department also have a lot of students, and the student may looking for some summer intern positions, may looking for some permanent job position, and we do have some openings for the next year and in this area. I know that's a very long introduction for very long introduction for myself and for Eli Lilly and the company and for the team, and also particularly talking about what we are doing in the research areas and how the potential collaboration point to work with these teams. With that, I want to kind of our team doing our team is more like the research group, and we are really trying to hire the top-notch the student, the candidate. Some of actually maybe four of my team members are from the universities, and previously is either assistant or associate professor, even for the tenured associate professor. They joined my team and to doing the research. I would often talk with other team members doing the analytics. They often have a different level to doing the analytics. It's like the sports. I have 2 daughters. They are one of my daughters was in the elementary school, so they have a elementary school level the football. They call it flag football. So the middle school level, high school level, college level, the football, and also the NFL level, similar like doing the analytics. They also have a different level, similar at the kind of the league of the sports. And my team is trying to hire this NFL level of the analytical people. So that's why kind of, and we are doing a lot of research, and also we publish a paper. We also publish papers to support the drug development. So that's kind of what our team was doing. So today I'm going to talk about a topic is that we call a direct learning algorithm for individualized treatment recommendation. So the reason we talk, because this is only one of our research, only one of our research we want to, the most recent research, we just want to give people a little bit of flavor on what we are doing for here. The background of this topic, I have a kind of twofold of this background. The one problem is why we are interested in this problem. This problem is related to the recommendation system, the individualized treatment recommendation, which drug is the best for this particular group of patients. But the treatment is only kind of the concept of treatment can be more abstract. We can give any of the recommendation on the intervention. So the intervention we recommend the patient to do some exercise. We recommend the patient to take some drug at a certain time. So all this belongs to the recommendation system based on the individual situation. So in general, this area it belongs to one of the recommendation area, and also belongs to the personalized solution, because finding the heterogeneities of the patient population, which patient is suitable for which treatment solution, why the patient is different. That could be one very interesting area for pharmaceutical company. And actually, this is also very general for the general public health. And so one interesting question, I know some people on the phone were doing the research on the COVID-19. I saw Steve was, when they introduced at the beginning, they mentioned this COVID-19 research. And so the very interesting question is why some people have this moderate kind of the reactions of the COVID-19, and why some people is asymptomatic, why some patient is have very severe reactions. Certainly age is one very important factor. What else? Is there any, among all of these factors, what is the modifiable factors? What is the non-modifiable factors? And how can I, if I got this COVID-19, how can I kind of prevent myself from become more severe the situation? All these topics belong to the heterogeneity and related to this personalized solution. How can we really understand each individual patient situation to provide some solution to optimize the treatment? So this is the kind of the topic related to what I'm talking about for here. And based on this, we do some of the research, and I'm going to share some of the research. I'm going to share some of the research for this talk. So let's get started. I know I have a very long introductions. I hopefully kind of people through this can better understand the background and so what we are doing. Now the second part, the second part of my seminar presentation is really dive in some of this research, and kind of maybe there's a more mathematical theoretical driven. And through this, I just want to show one of our research what we are doing, and to kind of show some of the work that we're really proud of and achieved a good result. As we talked about before, and many of our research in the pharmaceutical research these days have been in the stage is precision medicine or the personalized solution. So the one size fits all solutions have been the old paradigms. And how can we truly believe if we want to further improve the patient outcome, and we need to be individualized solution, because once we can, the only way we can achieve the maximum benefit for certain population is to maximize the benefit for each individual patient. The few topics people may heard about is subgroup identification, personalized solution, individualized treatment recommendation, contextual-based recommendation system, clinical decision system, or any personalized intervention algorithm. Ultimately is kind of talk about one thing is to there are a lot of research in this area and our topic is to focus on this. This just provide a high level of the introduction on the literary review in this area. During the past maybe 10 years, I myself is also very involved on some of this research, and I list out a few papers that in different category that I also I'm got involved on searching some of the solution, including some Q learning, A learning, or O learning method to use how can we better use the machine learning to solve this problem. But in general, the problems belongs to one category called the reinforcement learning category. So in the large scale, the machine learnings have three categories. One category we call the unsupervised learning, supervised learning, and reinforcement learning. You can think about unsupervised learning is, for example, the clustering analysis, principal component analysis. In that case, we only have the so-called features or covariance information, or in mathematical term, we only have the X, the covariance information. So we want to make the people in a different group, and we want to that's the unsupervised learning. The second group is the supervised learning. Supervised learning is like a classification. We have the label, we have the X. We want to do the classification. That belongs to supervised learning. So these days, when the machine learning is continue involved, that's one area, the very important area, called reinforcement learning. The reinforcement learning have three components. As you remember, unsupervised learning have only one component, covariance information have only X. Supervised learning have two information, label and X. So there's X and Y. So our reinforcement learning have three components. The situation, action. The situation is X. The action and the result. Situation, action, the result. So under the situation, what action we took, what the result looks like. That's called reinforcement learning. It's like humans, mammals, animals. We all learn from its appearance. What do we mean by its appearance? Our appearance exactly contains these three components: situation, action, and the result. Under the previous situation, if I took some action, I have a better result. In the futures, I will reduce the activity. But on the other side, if I have at a certain situation, I took some action, I have a good result. So in the future, under a similar situation, I will do more. I will do more of these actions. So I will achieve better outcomes. So that framework is called reinforcement learning. So machine learning has three components: unsupervised learning, supervised learning, and reinforcement learning. So what we are doing, this methodology we talk about for here is a personalized solution. Either belongs to framework of reinforcement, either belongs to work or reinforcement learning. We want to use our learn from the previous experience and to come out a better decision rule. This decision rule can helping us to drive for better outcome. In our patient situation, if I learn for some subgroup of patient, I need to use the treatment one can get better outcomes. For those group of patients, you have age, gender, demographic values, give a treatment one can have a better outcome. Learn from previous data, so I can come out a decision rule. For the similar patient as like those patients in the future, I will give a treatment one for those group of patients. So that is how the algorithm becomes smarter and smarter. That's one of the very popular approach called reinforcement learning. If people familiar with this trend of the AI machine learning, and during the past, maybe started from 2016, and Google DeepMind be the best human being and play the Go game. So that in general, that framework is a reinforcement learning framework. So the place board game is under the composition of the stone. How can I put my next stone that can maximize my benefit, maximize my chance of winning the game? So that is in the framework of reinforcement learning. If you read the paper, the science paper, and published by DeepMind from David Seaver's group, that's talk about how they use the reinforcement learning algorithms to make this happen. Our motivation let's start from the most simple case, the motivation example. And we talk about if they give treatment 1 or treatment 2 for this recommendation system, the decision systems. If we talk about the binary decision system, A is equal to -1 or +1, the continuous endpoint, and Y and as a reward. So I will take some action, what the reward looks like. And start from most simple case, the randomization kind of ratio is kind of 0.5, and we can always build a model in this way. Y is equal to the beta 0 in the overall mean effect. GX, the GX is a kind of this prognostic effect, so which means it's irrelevant for the treatment. When they always have the trend of X and the kind of this GX can be non-parametric. This GX can be non-parametric. And the dx times A, the D is called a predictive effect. So this can really differentiate for the treatment and plus some epsilon plus some error terms. This model, actually the model is very general because the G and the D are non-parametric models. Once we believe the mean, actually for all of these binary treatment models, we can for the mean model, we can always model the mean as a beta 0 plus GS plus DS times A, because the beta 0 is kind of overall mean. After I put out the overall mean, I can estimate for both of the treatment group what the GS looks like, and I can pull out this DS.
Sorry, Haoda, sorry for the interruption. We cannot see the slides change. Did you change your slide?
I'm in the slide 5.
But from our screen, still show the slides 1.
Oh, really?
Now it's become 5.
Okay, I think maybe because the full screen, full screen problems. Okay. So I just reviewed this one and also the review some literatures. Now I'm in this slide 5.
Okay.
So you can see the slide 5, right?
Yes, we can see it now.
Okay. Now I may not use the full screen mode. Just share this one. So this will be the general forms. This will be the general forms of all binary treatment models. The question is for all of these personalized solutions, where our goal is to estimate the DX, the treatment by covariate interactions. So once we know the DX, then we can know how we can give the treatment of the patient. So if people in the area and do a lot of analysis and to estimate the DX, the treatment by covariate interaction will be the key problems for binary treatment decisions. However, if I have binary treatment and we want to figure out what the subgroup patient should receive the treatment 1, what another subgroup patient should receive treatment 2, that's estimate the DX. So I also want to point out for here is the equation Y is really the general forms. We don't want to talk about we don't want to solve some small problem, be only specific for one solution. We want to solve the problem in general enough. So the equation Y is equivalent to this general form. This H can be a non-parametric functions and H and X and A. So how can we solve this? So first we have some very interesting result. If we use 2 times response Y times A, we calculate expectation. That will be equal to 2 times DX. Because the beta 0 and the GS is the population mean levels, kind of the population mean, overall mean, and the mean value response to X. Now if the A is a positive 1 and negative 1. Now, if I use, you can imagine if I use A times this Y and some part is the positive, some part is the negative, I will cancel out of beta 0 and the GX. But A will become A squared. The A squared, negative 1 squared and the positive 1 squared will still be equal to 1. So the DX will be kind of left for here. By doing and also this DX have very good causal interpretation. Causal interpretation, we talk about potential outcomes. That means if the patient takes treatment 1, what's the difference with the same patient X to take treatment 1 compared with take treatment negative 1? This is still equal to 2 times DX. So they have this causal interpretation, and to measure the treatment difference. So that motivate us to think about one of the estimation equation. The estimation equation will come out with 2 times YA minus some fx. We want to search this f to minimize this equation. So this f will be the estimator of this dx. If this f can have estimator of this dx, or like actually 2 times dx, then we can use this fx to helping in the personalized solution. Now this gives us some of the motivation is, instead of to use many of our models, they only use the original y as a model, but now we come out a pseudo outcome. The pseudo outcome is y times a. Once we come out with pseudo outcomes, we can build a model and to kind of estimation equation, so we can estimate our f. So we can minimize these equations. This f will be suitable for personalized solution to better understand the subgroup. So that's the original motivation. The question is, how can we from here to go deeper and deeper? What other questions we haven't been answered? So for example, what if the expectation of A equal to 1 and the randomization ratio is not equal to 0.5? How about this kind of this probability also depend on X that become observational study? Many of our real world settings in the observational study, for which means the treatment assignment is not a flip of coin. The physician make the treatment decision based on the patient situation. So in that case, we collect those data. How can we handle the situation? To make it, to make that happens, and we are talking about this binary treatment continuous endpoint. Right now, I'm in the slide number 8. So we start from this kind of potential outcome framework. So what's the value for y for the patient? If a patient X, if I'm a patient X, I take the treatment 1, what's my benefit looks like? Minus the what if I take the same X, but I take a treatment minus 1, what my benefit looks like? Now I calculate this difference. If I calculate this difference, when the randomization is not depend on the covariance information of X, then we can calculate the left-hand side will be equal to this equation, YA divided by pi AX. So remember, in the previously, this denominator is equal to 0.5, and then that y, the 2YA is kind of equal to the treatment difference. By using a similar idea, now I can also build up an estimating equation, YI divided by this equation minus fx. Then I can search this fx. Then at the end of the day, this fx will be the estimator of this treatment difference. Then this fx, I can use that for the personalized treatment, the treatment decision to search for the subgroup. Now, by come out this pseudo outcome approach, we kind of solve these binary treatment problems. The pseudo outcome is, instead of fitting the fx, doing the least square estimation with the original y, and we from original data, we do a one further step to calculate the yi ai divided by the probability of ai condition xi. This denominator can be estimated by the logistic regression, and after that, these whole things come a new number. This new number can come out from our data. These new numbers will be a pseudo outcome. It's to replace our original y as a pseudo outcome. Then I can build a regression model. I can build a model with fs. I can use a linear model. I can use some non-parametric model to fit these pseudo outcomes. So I can get my estimation. This equation is also relevant to the bottom equation. This actually the exact same kind of but the bottom one I moved out for this pi ai xi outside of this kind of bracket, but and kind of at the same time, kind of you can do some little bit algebra and to calculate why there's a 2 for here and what kind of why instead of the square, why this kind of why this like this. The purpose for here is the denominator of this guy. The denominator of this guy. Sometimes the value could be very small. Sometimes it's not stable. And if they're only doing the denominator and a pseudo outcome, if I moved out from this, and so the whole equations, the calculation will be more stable. So this technique is not new. This technique is have been used for the causal inference field, and this some stabilized, they call it stabilized kind of calculations on the causal inference field. So we just borrow this idea, and we can make this become more stable and to benefit for the calculation. But from mathematically, the top one and the bottom one are exactly the same. Of course, and fitting the perfect data is not fitting the perfect data. Is not kind of the only purpose. If people working on the machine learning, if people work on the machine learning area, they know some problems so-called overfitting, and they need to add some regularization. So we can certainly to add some regularization like lasso estimators, and there's some other approach we can add it on this to make this penalize the function to make the variance and the bias trade-off. Similarly, we can also use the non-parametric approach and to extend this in the reproducing kernel Hilbert space. Now this kind of give a, now if we go back to slide number, if I go back to kind of slide number 5, this original question is the binary treatment. How can we handle this? And we come out this, there's a pseudo outcome approach, and then kind of we extend this to the kind of this observational study. We solve the binary treatment problems. But the question is, our major contribution for here is, that's 2 treatment, but how about the multiple treatment, 5 treatment, 6 treatment? How can we handle this? And that kind of extend this from the binary treatment with A can only be equal to -1 and +1 to multiple treatment is not a trivial case, and so that that's way there are some of the traditional approach like one versus all, one versus the rest. Those approach, the ad hoc approach, may not be is not optimal approach, and that's why we are thinking about how we can extend the algorithm from a binary to a multi-category cases. One of the challenging things is this equation depends on A is equal to +1, A is equal to -1 on the slide number 8. It depends on the -1 and the +1 structure. But how can we extend this to a 3 treatment, 4 treatment? And we also want to solve the problem elegantly. So how can we do that? Also, we want to, the result also interpretable is how can we do a better job on this? To make that happen, we introduce some mathematical tools to make this happen. The mathematical tool, we start from the this is kind of simplex action space. The simplex action space is like below. Suppose we have previously have 2 treatment, like the k is equal to 2, k the number of treatment or the called action, and if the k can be equal to 3, k can be equal to 4, and we start to form these basics. This basics is in the k -1 dimension space. We form a unit ball, and we have these vectors and equally spaced on the unit ball. To be specific, when the k is equal to 3, so k -1 will be the 2 dimension in the flat plane, in the 2 dimension. In the 2 dimension, I will put 3, my 3 basics. The basic is W1, W2, W3. These 3 basics are uniformly spaced on the unit ball. You can imagine a circle for here. The circle is a 2-dimensional unit ball, and W1, W2, W3. You can see any of these two angles, W1 to W2, and W1 to W3, and or W2 to W3 have exactly the same angle. Similarly, when the k is equal to 4, k minus 1 is in the 3 dimension. In the 3 dimension, you can think about the ball. In the 3 dimension ball, and where the basics is W1, W2, W3, W4. We form these 4 bases. You may ask me the question, why need to use the basics? And these basics, how these basics, this simplex action space is related to what we are going to do. And in the multi-category cases, I will answer those questions. So here is the property of these basics. And this equation is coming from a mathematician. Is for any number of this unit ball, the basis, what the mathematical form looks like. This form looks complicated, but the geometry interpretation is very simple, and their properties and added all these basics, they will be equal to 0 because they point out in the different direction that symmetric. The adding together is equal to 0. Each of these have a unit. Each vectors, the basics have a unit value is equal to 1. Any of these two have the same constant, have the same angle, and this angle from 0 to pi. Now the question is, how can we use these basics to solve our problems? We start come out this instead of to calculate the difference, and we know we want to calculate this potential outcomes because that is our ultimate need. So this equation starts from ultimate what we need for the personalized solution. If we think about the personalized solution, different patients have a different treatment or side different treatment for the patient. What's the ultimate what we really care about? So if you can imagine for myself, if I go to clinic, I take treatment 1, I survive for 1 year, I died. If I have an opportunity to coming back, I take treatment 2, I survive for 1 week. After taking treatment 2, I have really adverse event. I died for 1 week. So I can coming back to take treatment 3. If I take treatment 3, I survive for 5 years. Now, at the moment, of course, each of us have only one life. What we are talking about for here is a hypothetical case, is a parallel life. But the question is, if I do have the parallel life, I can take the multiple treatment. And when the moment I make the decision, which treatment I need to take, of course, I need to take treatment 3 because the treatment 3 will give me the longest life. Similar for here, this equation is exactly talking about the same thing. If I have the potential outcomes, if I'm a person X, in front of me, I take treatment A, I have a different choice. When K is equal to 1, K is equal to 2, I can take the different treatment. As I take different treatment, I have a different outcome of the Y. So if people ask me which outcome I need to take, of course, I need to take the treatment that can maximize my benefit. So that's what this equation talks about. What the treatment can maximize my potential benefit. The D0x is my this decision function. By some algebras, we can do some of the calculation by introduce our basics of this W. This basics of this W. If once we introduce this notation, and so the numerator, the first line will be equal to the bottom line. Now, there's some derivation. We will not go through this derivation. The first line is added this ck minus this ck, and doing some of the calculation. And after that, this 1 will be equal to, because each of the norm is equal to 1. So wk transpose times wk will be equal to 1. And this ck different two vectors there, if they are not exactly the same, so that will be equal to this ck. And how this comes from this line, and we combine this together, we can get this equation, moved out of this wk outside of this equation. We can get this one by the definition of these notations. And in the end, the whole thing, this interpretation of yw divided by pi ax conditional x, we can define this as a function of f0. So in that case, by this definition, we can estimate this guy to maximize the benefit. So there's some derivations. Now suppose you already got your I will talk about in the last few minutes, talk about how we can estimate F0. Suppose you already have F0, how you use this F0 to estimate which class it belongs to, and they are using angle based. So that's how we use our basics. If a W1, W2, W3, and we have this F as estimated F hat, we just simply to measure. So which one have a smallest angle? The smallest angle will translate to largest inner product. We just measure which angle have a smallest angle. Then in the graph B, you can see this F have a smallest angle with the W1. So I will recommend the treatment 1. The W1 is the corresponding treatment 1. I will recommend the treatment 1 for the first patient, so on and so forth. So that's how this model interpretation. And ultimately, what we estimated is this the treatment difference between one group go to another group. So that's how this mathematically they're equivalent. So which means what we estimate is really estimate the treatment difference between one group over another group. How we can estimate our F? So we come out some of the estimating equation. This estimating equation is just follow the previous what we discussed and got this solution. And certainly, we also talk about the penalization. We can add the penalization in the end. So there are some of the different type of the penalization. For example, if we prefer this B have a low rank structures, and if we prefer this B have a sparsities, and we can add the different type of the penalization to balance the variance and balance the trade-off. Like in any models, you always see have a model fitting component loss part and the penalization smoothing the penalization part. This follow the general machine learning ideas. Certainly, what we just talk about Y is a continuous endpoint. People may ask, so what if Y have a different endpoint? For example, if the Y have this binary outcome, so what if Y we have this time to event? The time to event outcomes. So the following 2 slides will extend this to a binary outcome. We change the different loss functions and make this happens. And we also extend this to a survival outcomes and to handle this time to event data. In the end, there are some of the theoretical result. One important theoretical result is we call the features consistency. The features consistency means when the sample size, when we collect more and more data, is really our treatment decision, the recommendation rule will converge to the true optimal result. So the answer is yes, and so our result have these theoretical properties. And I'm not going to show some of the simulation and the real data analysis, and our result already published in the early this year in the JASA, Journal of American Statistical Association. This is one of the top statistical journals, and so you can take a look on the simulation result and the real data analysis. In general, I'm really proud of the solutions. So what I did to make a summary, and in this particular technical part of my presentation, and we talk about this direct learning approach to how we can make this approach as a personalized solution to maximize the patient outcome. And this solution is we have only one estimating equation. It's simple, flexible, and this can be really generalized. This if people interested in to generalize this in the deep learning approach, and we have the loss function to target loss function. We just simply replace the fx as the deep learning models, And in the end, we have this deep learning model, have this final loss functions. You can calculate the derivative. You can calculate use a back propagation to calculate each of the derivative, the gradient of the parameters. And you can fit this model. And if you are interested in the random forest or tree algorithm, this model can easily to extend for other framework. So kind of in the futures, and right now we also work on making this model for like dynamic treatment regime or some other to make in the sequential of decision makings. So with that, I want to kind of conclude. With that, I want to conclude my presentation. I want to give this kind of let the people, in case people have some other questions to ask. The question can be general, like a question in Lily, question in our team, and a question in this talk in particular.
Yeah, thank you, Haoda. Very nice talk. So any questions for Haoda? So Haoda, I have a quick question. So yeah, it's a very neat work, framework, and also your detailed solutions. So my question is, for this type of approach, especially when you use the observational data, you actually need to assume all of the confounders are collected, right? Yeah. Otherwise, the inverse probability weighting or probability score may not work properly. And in terms of a causal inference, there is another sort of approach, which is using the instrumental variable. But this approach probably is a sort of a mainly used by the econometrics or econometrician. So I'm wondering whether you know any sort of example of using some instrumental approach to discover the treatment effect of a drug.
Okay, that's a very good. That's also very good question. So there, in general, to handle this, they have a different approach. For here, we need to assume there's some assumptions, including no unmeasured confoundings, positivity assumptions. That's a few assumptions we need to make. That's a kind of standard assumption to make this model works. And if the data is from randomized control trial, this will automatically satisfied. But from observational study, we always need to make such assumption. And to make this problem in the more robust, we can even think about the doubly robust solution. But but there are some people may ask, is there any some other different school of thought from the instrumental variable to handle the causal inference? But on the other side, the instrumental variable is also assumption based, and also the assumption is not the unverifiable assumption. They need to come out some of the variables, and this variable we assume this variable is the instrumental variable. So in general, kind of we, for not many problems, they can come out this instrumental variable. So that's why in the pharmaceutical companies, and this instrumental variable approach is not that popular, because first part, this assumption is also not verifiable. The second part is not every problem people can very intuitively to find interpretable instrumental variable to help in this. So that's in general the kind of the instrumental variable approach is not that popular in the pharmaceutical industry. But on the other side, regarding the first question and the unmeasured confounding, they're increasing interest in how we can better measure the unmeasured confounding. So you can imagine this is kind of, I think one professor from UPenn was doing a lot of research in this area. In general, if there's no unmeasured confounding, so kind of to build the model, the logistic regression models, and they can very well interpret the treatment assignment. So the difference from how good they can interpret the propensity score with some additional variable, they can measure the distance of the unmeasured confounding, how much of the degree of their unmeasured confounding. So there's a lot of interesting work have been coming out in this area, and we are also very interested in that direction.
I see. Yeah, thank you. Any other questions?
There's one I was just thinking of as you're talking about this. Is this kind of analysis, you might have said this, is this kind of analysis also used during clinical trials to dictate possibly shifting patients slowly from a control group to an experimental group, of the experimental arm is working very well?
That's the excellent, really excellent questions, and it's also kind of the change the paradigms. Let me give the big picture and also narrow down this. And so some of the large company, like even for the Google, we talk about the Verily, and they're also very interested in this type of the approach. So they mentioned that right now there are so many of the drugs, and for example, there are almost 100 drugs treating the diabetes. So is that more value to develop a new molecule entity, develop a new drug, or they can develop the algorithms. This algorithm can better use for the existing treatment, for give the treatment. So which one is more valuable? You can imagine, we can even imagine a randomized, double-blinded randomized control trial. So the traditional double-blinded randomized control trial is compare treatment A versus treatment B. Have these two treatments, why the patient randomized group A give treatment A, otherwise give treatment B. But talk about for the future, maybe the trial is still randomized control trial. If they randomize for the treatment A, they give a treatment A. But if they randomize for another arm, after this randomization, apply these personalized treatment algorithms for different patients, give a different generic drug, give the existing drug. And in the end, we can still compare these two arms, which have more benefit. Maybe in the very near futures, to leverage the existing generic drug, but with the better algorithms, they can generate more benefit compared with the new molecule entity. So some company was thinking about this, and so that's a very interesting paradigm change. So instead of develop a new drug, let's develop the algorithm. This algorithm can better leverage the existing treatment to generate more benefit. I'm thinking that's a tipping point. When some of the disease, there's only one or two treatment, to develop this algorithm may not be so beneficial. But on the other side, when some area have a lot of treatment and the patient either different with one over another's, kind of the very heterogeneous of the patient population, treatment solution is big. And in that, for those population, for those kind of this, when they have more and more drug on the market, the algorithm will generate more and more benefit. Lilly is also very interesting because Lilly is a large pharmaceutical company in diabetes area. We have multiple treatment. How Lilly can leverage this type of algorithm to leverage, instead of one drug, to leverage our portfolio to make our Lilly become more competitive compared with other company if they can only provide one or two drugs.
An excellent point, Haoda. So on that encouraging note, let's thank Haoda again for a great seminar, and give him a few minutes to prepare his thoughts for the meeting with graduate students, which is now only 8 minutes away. So thank you everybody for attending. A great attendance. We hit 41 participants today, which is a good number for a virtual seminar, and something you should be proud of, Haoda and you, Sijian. So thank you again. I'll look forward to seeing everybody. Sagar and I will look forward to seeing everybody on Wednesday, the 18th of November, for the first installment of how the SARS-CoV-2 proteins evolved during the pandemic.